Today we have with us Dr. Alex Kwan from Dr. Alex Kwan is an associate professor of psychiatry and neuroscience at Yale University. He was born and raised in Hong Kong. He received his BSc in engineering physics from Simon Fraser University and a PhD in applied physics from Cornell University. At Cornell, he developed non-linear optical microscopes in the laboratory of Watt Webb. For his postdoctoral studies as a Croucher Fellow at the University of California, Berkeley, he worked in the laboratory of Yan Dan, where he studied cortical microcircuits. Then, in the year 2013, he joined the Yale School of Medicine as a faculty. The research in his lab focuses on mouse medial frontal cortex. He is interested in how cortical circuits enable flexible decision making and how the dendritic dysfunctions underlie neuropsychiatric disorders. Also, his lab works in developing and applying optical tools to record and control the neural activity in the behaving mice. With that small introduction, the title of his talk is Visualizing the Rapid and Long-Lasting Actions of Drugs in the Brain. He will be talking about his lab interest in determining how the brain responds to the psychoactive drugs. Now, being from a physics background myself, I find Dr. Alex Kwan's journey very interesting, as I personally feel that sometimes when I'm focusing on the field of neuroscience, my experience with physics overlaps with a lot of things that Dr. Alex is doing in his research. And so I'm really very excited for his talk. So with that short introduction, I would like to introduce and welcome Dr. Alex to kindly present his talk. Thank you so much for joining us, everyone.
Try to share my screen. Yeah, is it showing?
Yes.
Good. Well, yeah. Well, thank you, Ruth, for the introduction. That was very kind.
Thank you.
Yeah, I think I have a physics and engineering background and switched to neuroscience, and I think it's quite valuable to have different perspective in the study of neuroscience. I think we each bring different skills and expertise to the table. So yeah, as Ruth mentioned, it's early morning here. It's about 7:30, I think. So I'm usually not a morning person, so I hope I keep my energy up. I have already drank a few cups of coffee, so we should be good to go. But I'm quite glad to tell you some of the works that we're doing in my lab. I want to start with this slide that is a bit more broad in terms of all the things that we're doing in the lab. I want to one of the actually other big thing that we are interested in the lab is studying decision making, which I'll not talk about today, but we have a number of papers and projects trying to understand how animals make adaptive decisions. So these are decisions that are made in environments that are uncertain and unpredictable. We're also more interested recently in value-based decisions, which is how they make decisions based on their past choices and actions. So that's not something that I'll talk about today, but if you have any question on those areas, I'm very happy to answer via emails or later. Instead, what I'll talk about today is our second focus in the lab, which is trying to understand drug function, in particular in relevance to the dendritic function and also the dendritic dysfunction in the frontal cortex. Mostly I'll talk about ketamine work. I'll touch upon a little bit in terms of our work on models for neuropsychiatric disorders. And then underpinning all of those studies, I think, is a appreciation and use of optical imaging methods. So I'll show you plenty of data using mostly two-photon microscopy, but also just generally optical methods, some of which we're also interested in developing to try to study these problems. So I'll start by framing the research in the context of depression. Major depressive disorder and mental illnesses in general are obviously very debilitating and a major problem in the world. Within the US itself, about 8% of the adult population have at least one episode of major depressive episode. So that's actually pretty staggering, right? So if you know 12 people around you, one person will have at least experienced one time depression in their lifetime. About half of the people there have very severe impairment. So by that, I mean, it's so debilitating that they cannot function properly, whether going to work or maybe even in their daily life. And then this is also twice as likely for women relative to men. One of the issue with depression is that also about the mental illnesses is that there are medications, but they are very slow and they work for too few people. So the common first-line treatment would be SSRIs, such as Prozac, which takes about 6 weeks to reach a response. So that's quite slow, especially when you consider that depression can come with suicidal thoughts. And that's something that you want to correct for right away. And then even with multiple rounds of treatment, about 30% of the people, about 1/3 of them, ultimately just do not respond. And once you're getting off the treatment, a lot of the people also relapse and go back and have problems again. So it was quite exciting in the US last year. The Food and Drug Agency, FDA, approved this drug, which is based on the compound esketamine, or a related compound ketamine, which I'll talk about. It's a nasal spray. It's a new agent, a drug that can be used to target and fight depression. Although you can see here also in the one of the premier newspaper in US, the New York Times, also mentioned that even though this drug has shown promise as an antidepressant, its properties are still not very well understood. And I think that's quite true in the sense that, you know, the clinical trial showed that this drug worked, but the discovery of the drug, which I'll talk a little bit about even, is quite serendipitous. So it was discovered by accident, and we don't really know why it worked. It was not found, I mean, there's some motivation, but it's not really discovered, I think, in a very rational way. So the idea is that if we can know more about the properties, like what this drug actually does in the brain, then we might have a better chance on improving the drug or finding other compounds that might have similar therapeutic properties. So, I mean, there's no complete lack of idea how ketamine works. In fact, there are a lot of models on how it works, right? We just don't know which one is right. I think one of the more prominent models is the neurotrophic model for antidepressant action. So consider here, you have a neuron, this is brain cell, here's a cell body, and you have the very elaborate, exuberant dendritic tree. And obviously you have the dendritic spine that goes on the dendrites, and which would connect with axon and form the neuronal connections in the brain. The neurotrophic model basically says that stress potentially weaken the synapses, whether it's structurally by completely eliminating them or weaken the synaptic strength. And then the antidepressant then may act to counteract that. So what I mean is like it might increase the number of spines or strengthen those synapses. It's a fairly straightforward, I think, simple framework. And then this might preferentially act on certain areas in the brain, for example, the frontal cortex or the hippocampus, to alleviate depression. And the molecular pathway that thought to be involved would be things that are neurotrophic factors or other kinds of synaptogenesis related pathways. And I think there's quite a lot of data supporting that these molecular machineries are indeed activated by antidepressants from work such as from Ron Duman's lab and Lisa Montega's lab and many other people, showing that likely that this molecular mechanism is involved in antidepressant action. However, I think one of the key questions that interests me, and I think it's quite important for the field, is that, okay, you can have plasticity and those molecules are important, but their molecules are almost everywhere. Like some of the factors like BDNF or mTOR exist in every single neuron and almost every single dendrite that you could find. It's very prevalent. But you might not want actually plasticity everywhere. You don't want your whole brain to become changeable and plastic. So how can this plasticity be directed to certain regions? I already mentioned that frontal region and hippocampus might be particularly important, and there's a lot of reason to expect that these drugs act on only some regions. How does it target certain cell types and or maybe even spines? So how can we make some of the circuit more plastic in order for the antidepressant to work, but not make the whole brain so plastic that you would lose things that you don't want to lose? So the idea that I'll try to present to you today, which is, I think, supported by some of our data, but I think remain to be further tested, is that we believe that these drugs might alter the membrane excitability of dendrites up here, and also modulate the calcium dynamics there, the signaling, which we think play a pretty strong role in steering that plasticity process to certain cell types, certain brain areas, and maybe even specific spines. So calcium is quite well known to have a very important roles in gating synaptic plasticity. So here, these are very classic work now from, I think, almost 30 years ago, showing, this is one experiment showing that calcium is essential for synaptic plasticity. So they have these light activated calcium buffers. So if you shine light onto the compound, it would then engage the buffer, and it basically chelate and take out all the calcium within the cell. So you can see that if you do something like LTP induction and then take away all the calcium, the LTP could still work. But if you just reverse that order in time and you take out the calcium first and then do the LTP, you see that it's almost completely abolished. So experiments such as these shows that calcium are really essential and necessary as a second messenger for synaptic potentiation, which now I think is almost common knowledge. And since then, other people have also mapped out very detailed these biochemical signaling pathways, showing again calcium have a very leading role in synaptic components, compartments, leading to activation of various kinds of downstream molecules that eventually lead to things like spine growth, what we typically associate with plasticity. So this is, I think, some of the motivation why we focus on calcium as an indicator for the dendritic excitability. So this is, again, the framework that I'll propose to you, and I'll show the data later. But just to frame, again, the whole discussion here, I think here is the classic neurotrophic model that has been, I think, fairly well supported with the neurotrophic factor leading to neuroplasticity. What we're saying is that it would be helpful to consider dendritic excitability and calcium, which we know are essential for driving these signals. And then as a result, I think the implication is that these excitabilities can be quite strongly regulated by circuit interaction, so interaction between neurons as well as cellular interaction, which then might steer the plasticity towards certain location. And then obviously, all of this is driven by fast-acting antidepressants, such as ketamine. So this is the outline of the talk. I will first talk about some work that we use imaging to track the remodeling of dendrites. I'll talk about some other work trying to look at dynamical signal, calcium signaling in spines. I'll circle back to talk more about what we think are the implications on these the actions of the drugs on dendrite. And finally, I'll share some just ongoing study, what we're doing right now in terms of advancing this framework. So ketamine is a very interesting compound. It has many effects on the human brain. If you give a single dose of ketamine to a human subject, a healthy human subject, for example, you would get something like this, where initially, acutely, within an hour, within the first 2 hours or so, actually, but peaks at an hour, you have a very pronounced psychotomimetic effect. So, which means that you have perceptual distortion. You might start seeing things that you're not supposed to see. You also have some cognitive symptoms like impairments in your working memory. However, there's also a sustained effect. Starting about 4 hours, and now it's been extended up to maybe about 2 weeks, you have this effect where you actually get an antidepressant effect. So this is, again, in humans, so they have a battery of tests, which is like a questionnaire to determine your depressive severity. You can see that there's a pretty substantial, notable antidepressant effect that's sustained. You might think that this is relatively late. I guess it is true compared to when the drug is on the body. By the time it's about 70 minutes is the half-life of the drug. By the time the antidepressant effect kicked in, the drug has largely been washed out of the body. But still, this 4-hour onset is relatively quick relative to SSRI. So it's still a relatively rapid onset antidepressant effect. And the good thing is it's somewhat sustained for about 2 weeks. So we're interested in, again, how this antidepressant effect comes about. If you look at whole brain data, where to begin, there's good evidence in both humans and mice that the frontal cortex is a good place to start. This is a PET study showing where metabolically ketamine induces activation in the brain in human. It's pretty broad, actually, you can see. Definitely includes some areas in the frontal cortex as being a notable activation. This is what happens in the mouse. If you give the mouse a single dose of ketamine, you see that it preferentially activates the medial frontal area, including some of the dorsal area that we're going to study. But it does spare some of the other sensory motor areas that are more lateral in the frontal cortex. So one of the big proponents, as I mentioned, of the neurotrophic model is Ron Duman. Unfortunately, Ron Duman has recently passed away. So he was a very good colleague, very good friend in my department. And this was a photo I took with him 3 years ago. But he was one of the early champion of the neurotrophic model, and also have provided a good amount of evidence that ketamine could have important effects on dendrites in the frontal cortex. This was one of his study 10 years ago now, showing that if you inject ketamine into the mouse, I think in this case actually rats, you do see a substantial increase in the number of spines, again supporting the idea that it really can increase the number of spines to restore maybe some of those lost connections in depression. Where we come in is that we thought that, you know, you can see that ketamine increased spine density, that like there are a number of ways that this could occur, right? You can, for example, have an increase in the formation rate. You can have them form more often. You can decrease the elimination, or maybe both. And also what's interesting to us is the time course of the structural remodeling. How does it change over time? I think that's quite relevant if you think about the time course of the actual therapeutic effect of that drug in depression. Because right now here, what Ron did was more static, right? So he, I think, injected drug and then cut the brain out 24 hours later, and you don't really know that full time course. So as I mentioned, we are, quite invested in optical imaging technologies. So we have 2 2-photon rigs in the lab, and this is work done by a postgraduate researcher in the lab, Victoria. Now, actually several years ago, one of our first studies in ketamine in the brain. So she replaced the cranium with a glass window and then could image directly into the dorsal area of the medial prefrontal cortex of the mouse. She uses the Thywan GFP mice, which constituently express green fluorescent protein in layer 5 pyramidal neurons. And if you use a microscope in a live mouse and you go into the brain, into the cortex, this is how you would, what you would see. You see that basically the dendritic branches and arborizations, and you're taking a Z-stack going down. You can already see that, on the dendrite, you can see the spines quite clearly. And this is not, you don't see the cell body, which is too deep. And this is really just the apical tuft, the fanning pattern up on the top of the cortex, on the superficial layers. So I'll go through this quickly. This is already several years ago. We have a very simple design where we inject either ketamine or saline into the mouse. We're imaging, and again, the idea is to ask whether you have more spine that form, or maybe you have more elimination or less elimination, ultimately then lead to a change in the spine density. This is what Victoria found. She found that there was a pretty dramatic effect on the spine formation rate. You can see here for ketamine, spine formation rate clearly increased relative to saline. Although if you compare between the elimination rate, which is the empty square here, with the empty square here and the saline, there's really not much different. So there's really this divergence in the formation from the elimination rate, and particularly the increase in the spine formation, that seems to drive this relative increase in spine density in the frontal cortex. One other thing that we can do also with optical imaging that you cannot do just by doing histology is that, just because you have a spine doesn't mean that you actually have a synapse, right? So there are also these new spines, like Philopodia has not matured yet and doesn't have that machinery, including the postsynaptic density, to actually function as a mature synapse. It's thought that you need about 4 days at least for these proteins to get recruited and for the synapse to form to be functional. So one thing we can do in optical imaging is you can track these spines over 4 days or even more, and see these spines mature and maybe even grow in terms of the size to know that these are indeed can become mature synapse. And this is something that we found, a fraction of these spines, although not all of them. In fact, some of them do retract and are not persistent, but there is a good fraction of it that are persistent and in fact become bona fide synapses due to ketamine administration, suggesting again that it's not only just spine, you really do form new synapses. One of the somewhat unexpected result from here that we don't quite know how to interpret, but it was very striking, was not only we see increase in spine, but we also observe some retraction in tufts. So here is one case where you can see before, we can see a pretty prominent tufts right here of a whole branch that seems to disappear actually. And we can show that it is true because you can go up and down with a microscope to show that really this branch has disappeared, whereas everything else is intact. And this loss of some branches seem to be very specific to the date of ketamine injection, and it's not sustained. So it's really at the time of the injection. So summarizing this structural part, what we see is that we see some connection. So let me explain this picture here. So what I'm drawing here is a cartoon, right, with a bunch of dendrites, and the thin red and blue lines are the axon that you will imagine coming in. What I'm telling you is that we saw, spine formation, increase in spine formation. So you see more of the black spines here now. And then we also see some branches, tufts that could be removed, but these are more immediate of a retraction. So this led us to a model, at least structurally, where you have some connection being added while other connection being removed. And we think that this might lead to a case where you might start receiving more input from one region. Depending on where you add and where you subtract, you can tilt the balance of where the input may come from for the frontal cortex. This kind of idea obviously remains to be tested. So we're one of the MD PhD student in the lab right now is quite interested in looking at different input, for example, from the amygdala or from the thalamus, and see whether this kind of idea might be true. That maybe ketamine, yes, it does increase spine density, but maybe it does so in a more subtle way that it actually rebalances the input rather than just adding a lot of new inputs. So that's the first part, and I kind of try to go back to the framework here. We're studying ketamine, and I show you some evidence, along with a lot of existing work already, of neuroplasticity, in our case, increased rate of spine formation. But I want to get back at this key point here, which is the calcium and the dendrite, which we think also play a very important role in this process. So Farhan in the lab, Farhan was a postdoc in the lab. He now has his own lab in Singapore in Nanyang University. He injected viruses to express genetically encoded calcium indicators. So that way we can not only look at the structure of the dendrite, but also look at dynamical signals related to calcium level changes, calcium concentration inside the dendrite. So this is a kind of image that Farhan would see. You can see that the signal is very dynamic, both in terms of spatially, but also temporally. It's actually a very complex type of signal. And hopefully you can also see maybe some spine type of signal, but I'll also zoom in. Again, like we're imaging really the superficial area where the dendrites are, just the tufts. So you think about where calcium can come in the dendrites. There are also a number of biological processes, physiological processes that could occur. Calcium could come in, for example, when the spine is activated, when it receives synaptic input, like there's, for example, glutamate release on the presynaptic side. Then you have NMDA receptor open, and then calcium can come in, which would be very local. But you could also have some more global events. So these would be things like back-propagating axial potential, dendritic spikes that are actually regenerative current that would excite a lot of the larger chunk of the dendrite. So we can resolve some of that with the imaging. So here is a zoom-in version of that image I show you, and hopefully you can see sometimes that this spine, for example, is very clear, where you can sometimes see very localized signal, just that spine. Other times you can see the whole dendrite lit up. So these would be some of these local and more widespread events. So here's a still frame showing what I mean. You can sometimes see, again, just the spine lighting up, suggesting it's a local synaptic activation. Sometimes you can see a more widespread event. These events have different implications for plasticity. Some people think that this kind of dendritic spike is very important for plasticity, although obviously localized event, as I shown earlier with the biochemical signaling, can also activate the a cascade to induce strengthening. So in this talk, at least, we'll focus on the local influx, which reflect the synaptic activation. So we do a number of data analysis methods to regress out the widespread component and really try to convert some of these calcium signal to events. I tell you that we can do this to localize a local component, right? But you shouldn't take it just for granted. And then we also did some experiments to show that this is indeed true, that what we measure in that spine is really related to the local event. One way we can do it is to do two-color imaging. So here is also still two-photon imaging, but we injected two different genetically encoded calcium indicator. One is red, the other is green. We put the red in the dendrite and the green in the axon, and you can identify things that are adjacent to each other, that are opposing spine and axonal boutons. And if they are indeed coupled, then you would expect whenever the axon is activated, you would detect the same signal in the spines, right? And indeed, that is the case. So here's the bouton, here's the spine. They're not exactly directly correlated. Sometimes the calcium can go in the bouton, but there might not be a release, right? It's a stochastic event, but they are fairly correlated. And if we do an analysis between correlated adjacent boutons and spine pairs versus ones that are shuffled, we see that most of the cases about when they're adjacent, about 60% of the events are indeed correlated. So the majority of the event that we are imaging in the spine are indeed have a correlate on the presynaptic side. So they're indeed related to synaptic activation. So the design of the experiment is quite simple. It's very related to the ones we had before. Here we're just looking at calcium signal. We inject ketamine or saline, and then we see what happens to the calcium dynamics in the spines. What we found is a very notable and I think somewhat unexpected increase in the synaptic calcium signals in the spine compartments after the injection of ketamine. So here are two spines that we track in the same branch. And again, here, I think the strength of these type of studies is within subject design. You can look at the same animals and really go before, after. We could potentially, even look at days after, which we haven't done in this case. But you can really track the same spine and ask what happened to it. So here you can see that for the saline, there's not much of change, but then for the ketamine, you see a significant increase in the both the amplitude and the frequency of the calcium events, which we can quantify from hundreds of spines that Farhan has imaged. So if you see an increase in the synaptic calcium transient in the spines right here, there could be a number of reasons for why that is happening. One possibility is maybe these spines get more excitable. So you have an increase in the memory excitability. So for the same amount of input, it's really sensitive now and responsive. Or maybe you have actually elevated presynaptic firing. So you have more neurotransmitter being released. As a result, you detect an increase signal on the postsynaptic side. Or it could be both. So to try to isolate these possibilities and actually especially try to understand the postsynaptic side, Farhan did one additional experiment, which is to control the input. So previously, we were measuring just spontaneous synaptic calcium signal, just let the brain do its thing. But right now, if we can control the input, then you can try to start identifying whether it's the input that's changed or the output that's changed. And to do this, he used an electrode to excite the axonal afferents. So it's known that retrosplenial cortex have a pretty strong projection to the frontal area. So he artificially stimulate and control the input, and then at the same time measure the calcium signals in the spines in the frontal cortex. And what he found is that even in this kind of regime where you control the input precisely, you can still see an increase in the responsiveness, suggesting that at least in part what we observe is due to an increased postsynaptic excitability. So you can see here he inject repeatedly in the retrosplenial cortex, excite those axon, and you can see the increase in the responsiveness and the calcium signal in the frontal cortex. So I mentioned that this was unexpected to us. And the reason is that if you think again about how calcium can get in here, it could go in through NMDA receptors. But if you know anything about ketamine, pharmacology is that the ketamine is actually an NMDA receptor antagonist, so it should block NMDA receptors. So it's very unexpected that something that would actually should reduce calcium influx by blocking the one of the main calcium permeable receptors actually lead to an increase in synaptic calcium. One possibility we thought would be that maybe there's also additional cortical circuitry around it, and then blockade of other kinds of neurons, particularly neurons that project to dendrite that control these areas, the inhibition in these areas. might play a role. And you can think of a scheme where if you block the receptors, make these neurons less responsive, that would relieve the inhibition, specifically at the dendritic compartment, that could then actually raise excitability and lead to that increase in the calcium influx that we saw. So we look for evidence for this kind of change. And one of the good things about imaging is now there are a lot of genetic targeting strategies. So we also can put the calcium indicator specifically on these dendrite targeting somatostatin neurons in the cortex. Indeed, when we look at the cell body, as well as when we look at specifically at the dendrites in the layer 1, so these are going to be the input that goes on the dendrite, we see that there is a reduction in their activity levels, suggesting that this inhibition scheme is can be supported by just directly looking at the activity. Not only can we do that, we can also try to do causal perturbation to show that this pathway is quite important. So here we collaborated with Ron to generate this AAV-mediated strategy. So it's a Cre-dependent siRNA to knock down an NMDA receptor subunit, gluN2B. So we inject this AAV in there into the somatostatin Cre mice, then we can have a specific knockdown of the gluN2B in these interneurons. So you would expect that if you knock down the NMDA receptors here, the subunit, you would, well, first of all, you should reproduce some of the ketamine's effect. So you would reduce these neuron activity, but you would, and then also reproduce the effect in the synaptic calcium, but also you should render additional ketamine ineffective because you're already knocking them down. So if you additionally add ketamine, it should not do anything. So indeed, that's also what we saw. This is just a summary of many experiments, but this is really the key point where when we do the knockdown with this siRNA, we see that you can, and then you image now the spines and the pyramidal neurons, you can see that you can recapitulate that finding that we saw. You can see this increase in the synaptic calcium transient in the pyramidal neurons. And furthermore, if you add ketamine now, it doesn't really do anything. So linking this to depressive-like behavior, Ron's lab have used the same virus and then also tested a variety of depressive-like behavior using assays like forced swim tests and novelty suppressed feeding and so forth to show that this knockdown is also important for the antidepressant effect of ketamine in mice. Here, our work really focuses on the calcium signaling and show that there's a very strong immediate effect on the calcium signaling. So I do want to link it back to some of what people earlier have found in this area. You know, we're not the first one to show some disinhibition associated with NMDA receptor antagonists. But I think what we show here to you is a focus on the dendritic part, where we see this elevated calcium signal in the dendrite, decrease in their activity, these dendrite targeting neurons. I didn't show you the data. We also used electrophysiology measuring LFP to show that it also changes long-range cortical connectivity when you mess with this dendritic inhibition circuit. And it contrasts actually quite, I think, starkly with some of the current idea of disinhibition, which tend to focus on inhibition of the somatic inhibition, which would be PD interneurons. So these are these ideas is championed by people such as Abida Mohanam and John Lisman, showing that, and it consistent with a lot of finding as well. So if you have this inhibition in the soma, then it's associated with elevated pyramidal activity, which Beta's lab has elegantly show, as well as decreased fast-specking cell activity in aberrant gamma band oscillations. So I think one of the interesting ideas going forward and would be fun to test would be whether these type of disinhibition pathway act in parallel in the brain. Like, do they both of them contribute to the antidepressant effect, or are they separate? Maybe one of these are more important. It's not really clear. Also, do they act in parallel, or do they act in tandem? Maybe you need both of them, and maybe they are affecting each other, and they're both of the components are quite important in terms of the pharmacology of ketamine. Going back to the framework, so ketamine, so we provided some evidence that it induces elevation in the synaptic calcium signal, then that we think that this is quite important then later on to drive these biochemical signaling and neurotrophic factors that other people have already observed and widely reported with these drugs. But the important part is, these calcium signals are quite strongly regulated by the circuit interaction, in this case, the somatostatin neuron circuitry. So I think it's fun to think about what this means. So if you have these structural changes and calcium signaling changes, what does it mean in terms of the function of these antidepressants? One of the things to think about is where are these inhibition on the dendrite? I think there's good evidence from electron microscope that microscopy that the inhibition is actually on only some portion of the dendrite. In fact, it's also mostly on the shaft, so it's not on the spines. So this is early work from a Japanese group, the Kawaguchi group, showing that if you do electron microscopy and look at exactly where these synapses occur, you see the inhibition right here. These are symmetric synapses on the EM. You also see excitatory pathways, synapses coming in here. But you already see here, for example, you have a spine that is very close to a inhibitory synapse, so these could be pretty strongly influenced by this inhibition, but you also have spines that are quite far that would not perhaps be influenced by the inhibition. So you see a probably gradient of effects when you put on ketamine. So this is the idea that we have basically. We think that this inhibition could be quite localized depending on where those SST neuron synapse on the pyramidal neurons. Here is to show you one SST neuron. Obviously, you have many of them. So you have this place, you have this idea where now you have heterogeneity of where the dendritic excitability may be increased, and in some cases might be decreased by the direct action on the NMDA receptor on the dendritic tree. So one reason why this is important is we show that the calcium influx could be quite important for plasticity. This would suggest that certain parts of the dendritic tree might be preferentially experiencing this increase in the plasticity potential. We have some evidence for that, I think, when we look at the data that we saw, looking at the increase in the calcium signal, actually, we see that not all the spines have this increase in calcium signals. In fact, about 30 or 40% of the spines did not change the calcium signals much when you do this manipulation. So a fraction of the spine seems to be much more responsive to the drug. Another thing I think to think about is that if indeed somatostatin neurons are quite important for this process, the distribution in the brain is also not uniform. So you can have more in one area than the other relative to other kinds of interneurons, such as pyrobumin interneurons. And then so you could have a shift in the balance inhibition between dendrites and soma. And if you look at this PD to SST ratio, which other people have looked at, and we recently have also mined it based on the Allen Brain Atlas, you can get data of the number of transcript in the mouse. You can see this trend where there are indeed more, relatively speaking, SST interneuron in the frontal cortex relative to the bulk of the inhibitory neurons in the frontal area, as well as some of the more interesting like temporal area relative to somatosensory area and visual area. So this could perhaps explain in part why some of these drugs might have region-specific effects, particularly areas that might have more of these kinds of dendrite targeting neurons. And we have done some experiment along this line. Again, I have shown this image already in the mouse. It's known already that ketamine has a much stronger effect in the medial frontal area relative to, for example, the primary motor cortex. When we image the primary motor cortex, we also see that it does not have the similar effect on the synaptic calcium signal. If anything, after injecting ketamine, you see a leftward shift in your curve, meaning that you have less synaptic calcium signal. So that what I showed you previously about this increase in the calcium signal in the pyramidal neurons is quite region-specific to the medial frontal cortex, well, at least in comparison with the primary motor cortex. So I think that's a good point, and now talk about maybe some of the ongoing research that we're quite excited about. I've shown you some evidence supporting this framework, but definitely I think more can be done. Some of the things that we're interested in are neuropsychiatric conditions. So the thing I've shown you right now, a lot of the work has been done in wild type mice, but obviously you want to study this in the context of depression, or at least conditions that are relevant with depression. So it'll be useful to look at neuropsychiatric mouse models. We also are interested in other compounds. So does this generalize? Ketamine is just one compound. What about other compounds that might have relevant effects? And also, the places that we're imaging in dendrites is relatively small. What about, can we image more and look at other events like the widespread calcium events that I talked about? So in terms of neuropsychiatric condition, my lab has been also quite interested in chronic stress. postdoc Florent Bartlas, as well as MD student Melody, has published some works on in the lab doing social defeat. So here we have a 10-day regimen in social defeat, and then we have behavior as well as imaging. On the behavioral part, we develop a new sucrose preference task. I don't think I have that much time to try to explain this, but in effect, it's a head-fixed version of a head-fixed version of a sucrose preference task. And we can show that susceptible mice have a greatly reduced preference for the sucrose relative to resilient mice in these social defeat paradigms. We can do imaging, longitudinal imaging. Here we use a specific construct that allows us both to image dynamical signal from GCAM as well as static signal, so we can track the same neuron over days. We can do it almost every other day in this regime, looking at what happened to the same cell pre-stress, during stress, after stress, track their trajectory over time. Here to show you how it looked like every single day. You can go back to the same cell. And I won't, again, go too much into this study. It's published and you can read about it. What we found, at least when you image the cell body in this case, is that there are fairly heterogeneous changes in the brain through the cell. And we found that some cells seem to correlate with some of the behavioral changes in the sucrose preference. So they seem to track the changes in the behavior. I think going forward, it will be very interesting to now look at dendrites. So all of this that study with the stress in the cell body, it'll be very interesting to look at the interaction of ketamine with stress, but at the dendritic level. What happens even in the stress? How does it even do alone at the spine? And then what happens when you have them interacting? Another thing that's ongoing that we got quite interested in other compounds, psilocybin is a compound that recently received a lot of attention. It's a psychedelic compound, but it's also been found to have potentially some rapid antidepressant effect. And in the US, at least, it's been designated as potential drug breakthrough therapy, and there's ongoing clinical trial. But regardless of the actual, I think, utility, what excites me the most about these compounds is that they act on serotonin receptors, and they're just choke full loaded on the dendrites. So if you stain them here, it's a Pat Goldman-Rakic work at 30 years ago at Yale. You see that these receptors that these compounds add on are just really densely packed in the apical dendrite in the monkey prefrontal cortex. Same thing for the mouse work done by Brian Ruff. So they must do something very interesting. The dendritic excitability is something we want to study. And other people have also shown that these compounds have very striking effects or related receptors, striking effect on plasticity. Actually, this is quite a while ago, but very well-cited work from Vedita showing that, and in Ron's lab as well, when she was at Yale, showing that when you activate these 2A receptors, you can lead to changes molecularly, leading to increases in factors related to plasticity. More recent work showed that if you look at culture neuron, you can increase the structural plasticity. So we just got some of these compounds and we put them in mice. It's quite interesting. I mean, they have a lot of interesting behavioral effect. This is the very well-known head twitch effect if you give the mouse psychedelic. I've seen this a lot in different plots, but I haven't seen a lot of videos of it. So we took some videos of it. This is in normal speed. If you give the mouse drug on the left, you can see that it's very fast, the video, just 1 second clip. You can't see much. But if you slow it down now, you can see this very clear head twitch effect, where you can see the head going left and right. So these compounds already have very interesting just motor effect on its own. And you don't have to train the mouse to see this. And these kind of effects really relate to the hallucinogenic part of the behavior for the mouse. Let me see. So I have a couple more slides. I'll just finish it. I think, and then finally, I also mentioned some work trying to look at more widespread event. The motivation for that is that if you look at synaptic input, which we focus on right now, you're really limited to certain kinds of dendritic events. But if you go back to your textbook, neuroscience textbook, you know that dendrite can actually support different kinds of electrical events, local NMDA spike, tough potential, or even some more global events involving back-propagating action potentials. It'll be very interesting to distinguish them. We think they're quite important in terms of what it means for plasticity. So Neo, MD PhD student in the lab, is now working on improving the imaging methods. So we now in the lab have the capability to not only image a small field of view, but to image now multiple field of views. You can split the field of views and track the same dendritic branch. And then also to look at different Z-plane. So not only at the tufts out here, but also in the trunk. So you can again, try to really capture that whole dendritic tree to see if we can visualize what the drug does. We're also interested in some neurobiosensor, not only calcium, but you can now image things like glutamate and neuromodulators. So this is it, a summary. I tell you about the drug, which is very important for treating depression, but we don't know how it works. I've told you that it increases the number of spine, drive formation rate. I've shown you that it increases calcium transient, which was a bit unexpected. Show you some evidence that it does that through a circuit interaction with the dendrite targeting neurons. I've given you a model, which I think is somewhat supported. Some evidence again, but also still not quite sure yet, right? I'll show you that cartoon with the red and blue changes in excitability, but it really needs to be confirmed. And we're trying to develop some new imaging technology to try to really image that dendritic tree to pin that down. We could be wrong, but I still think it's quite important to know what is actually happening there. And with the ultimate goal that I think this kind of signature at the neuron and neural circuit and system level could be quite important for identifying new compounds that might have similar effects in the brain. So with that, this is my lab. I've tried to point out how they have helped on the various part of the project. Collaborators, Ron Duman, Marina Picciotto, Chris Pittinger at Yale, the psychedelics, we've gotten them for the Usona Institute, as well as I want to thank the funding support. So that is it. So I hope you enjoyed it. I'll be happy to take any questions.
Thank you so much, Dr. Alex. That was a very wonderful and insightful talk. Plus, personally, I'm really excited that you're asking such interesting questions and also working on developing the neuroimaging techniques and microscope techniques that are needed to study these patterns. And being from a physics background, I would say that it's microscopes and optical imaging is like the backbone of the field. Without these advancements, it's very difficult for us to understand what is happening inside the brain. So you are targeting these two things in parallel. It's really has given me a lot of motivation to now I know how to proceed in this field further with my background in physics. So thank you so much. And I would like you to take questions. So first, Manusmriti is asking, since calcium signals hold so much importance, to what extent, in your opinion, can the mitochondria and the autophagic processes be involved in this process?
Yeah, so I have kind of pitched this idea that the calcium comes in through the dendrite, comes in through NMDA receptors and voltage gated channels, and then go into the second messenger pathway. But as you pointed out, calcium obviously can have, well, not only can calcium also have other sources like internal calcium stores, and then downstream calcium could also be important for other things, like in mitochondria and other type of processes. So I think all of those are kind of somewhat unknown. I think I think those could also play a very important role, and they're somewhat understudied in terms of what the drugs can do. And also did not mention, but for the psychedelics, which we're quite interested in now, it also these compounds act on the serotonin receptor, which is G protein coupled receptors, also regulate calcium that way. So I think it's not going to be like a flow chart like simple like this. There's going to be, I think, more parts to it that we need to understand.
Thank you, Dr. Alex. Thank you, Manusmriti, for your question. The next question is by Kimi. So she's saying the functions of ketamine seem to be similar to the activity of acetylcholinesterase in terms of altered synaptic architectures, including spine formation, arborization, etc. Can we replace ketamine with acetylcholinesterase inhibitors to improve neurocognitive symptoms in Alzheimer's disease?
Yeah, so I think if you think about what we currently know, or think about neuropsychiatric disorders or Alzheimer's disease or neurological disorder, the general picture is that they definitely important for dendritic spines. Some of them increase, some of them decrease. Maybe schizophrenia is increased, autism is decreased, depression is decreased. It's a bit hard to make sense of it, right? And a lot of these drugs seems to also change the synapse number. So I think what we try to do is to ask more precise questions, like where are these synapse increase, and also what are the inputs that are then specific to these increase. I think that might be a more fruitful avenue to go forward. So you're right. There are a lot of drugs that could increase the number of synapses and strengthen synapse, but I think there should be more specificity to that because they do lead to different phenotypes. And these drugs ultimately do have different actions on humans. So I think, yeah, it'll be useful to ask, yeah, how these drugs differ in terms of some more details about what they do to synapses.
Thank you. So our next question is by Ankush. He's asking how different are the effects of SSRIs from rapid-acting antidepressants like ketamine? Is more dendritic spines building up seen for SSRIs too?
Yeah, that's something that we have thought about, and it's quite interesting because if you SSRI also changes serotonin. So I mean, I guess I didn't talk much about this. Is again just an idea, but you could ask, why some of these psychedelic might have interesting effect, but not SSRI, or you can even ask, why does endogenous serotonin in our brain, which also activate these receptor, they don't give us hallucination. I think those are actually very open questions. You guys asking a lot of interesting open questions that I think deserve more experiments. And yeah, it would be very interesting to do some kind of similar dendritic experiments, like looking at calcium signals, but with SSRI and see what happens. I don't know if these experiments have been done.
Okay, thank you. So we have Dr. Vidita who wants to ask you a question. So I would request Dr. Vidita to kindly come on the screen and ask a question.
Oh, okay, great. It did allow me to unmute myself. Hi, Alex.
Hi,
Nice to
Vidita.
see you. A quick question on whether you think the architectural changes are predominantly restricted to projection neurons, or you think PV and SST neurons will show similar calcium signal and architectural changes. And what are your thoughts on the persistence of these effects, especially given hallucinogenic compounds are associated with perceptual flashbacks? Do you think you might see very long lasting disruption of calcium signaling that may persist long after drug exposure?
Yeah, so I think that the time course is a very interesting question here. So for there are multiple parts of the question, right? For the calcium, what we're imaging is a more acute time scale. We're looking at calcium changes that happens within an hour or two. What we think is that that's a pretty rapid thing, and then eventually lead to this downstream cascade. And going back to the neurotrophic model, and also the molecular signature that you and others have shown, that are then important for the more longer term plasticity. We just think that it's kind of one of the first steps in this process that then lead to that long lasting change. And then on the other aspect, which is the changes for the psychedelic in terms of the distorted perception, that's an interesting point because psychedelic and ketamine, let's say right here, they have very different acute effect. So if you give it a human who have used these drugs, they can tell the difference very readily. In fact, even within some psychedelics, people can tell the differences quite readily. So they're definitely not equivalent in this case. And what they do to the dendrite and how they affect it could be quite different. So I guess what I want to say is they definitely also do not only affect right frontal cortex and hippocampus. And I think the short-term effect might be involved with how they affect different areas. So Chris Neal has a pretty interesting study showing that psychedelic can have direct effect on the visual cortex. So that's something that might be absent for ketamine and might be more responsible for the hallucination. And then for the ketamine, I also want to mention on the time course, what we think, this calcium, again, neurotrophic factor, I think is a longer time scale thing, right? But there's also the shorter time scale, rapid onset within 4 hours. So that could be something else as well that we don't quite understand. I think there's some suggestion maybe the lyra habendula. There might be also some direct effect on the strengthening that doesn't really quite fit with this framework as well yet. But I think, yeah, that is also another kind of important question and kind of unknown in the field.
Thank you.
Thank you so much, ma'am. We have our next question from Nasa Kumar. Ketamine is commonly used as an anesthetic and is associated with memory loss. So with reference to that, will the neurons in the hippocampus have an increased spine density?
Yeah, so ketamine, depending on dose you use, have very different effect. So the dose that I show is a sub-anesthetic dose. So you don't get the anesthesia effect when you give the mouse at about 5 to 10 mg per k. Instead, you actually even behaviorally, you also see hyperlocomotion. They walk around more. So it's very dose dependent. If you use a higher dose, so an anesthetic dose, then obviously you go into anesthesia and you have a decrease in neuroactivity. And Matthew Lockham's lab actually has a pretty elegant, I think, a Cell paper last year showing that if you at that higher dose, you also see very striking effect on dendritic signal. But what they see is a complete segregation. So it becomes very segregated and electrotonically distant. So they're electrically discoupled or uncoupled. So yeah, depending on the different doses, you can have different effects on the dendritic signaling. But here we're doing the sub-anesthetic dose, which is more relevant for depressive disorders.
Okay, so I'll be taking the last question for the talk, and it's from Sudipta Sarkar. For the very initial test of verification of action of ketamine for increased spine generation or decreased spine degradation, why do you think there was a drop in degradation both in the case of saline buffer and ketamine?
Sorry, can you repeat that question?
For the very initial test of verification of action of ketamine for increased spine generation or decreased spine degradation, why do you think there was a drop in degradation both in case of saline buffer and ketamine?
Oh, I see. You mean why is there? You mean in the spine density test?
Yeah, why is there a drop in the degradation?
Yeah. So that was we report what we saw. So it was a bit unexpected why even the saline animals seem to have a drop in the spine density or the spine formation rate in this case, but it lead to a drop in spine density, even if we give the animal saline. We think that might be a little bit of an artifact of the experiment. So first of all, we were using adolescent animals. So these are younger animals, and there probably still a little bit of pruning going on as the animals develop and grow older. But more importantly, I think, when we do these imaging, we are anesthetizing them every other day. And these are pretty long imaging experiments to try to image the dendritic tree. That could itself be somewhat stressful to the animal. So yeah, we do see some changes in spine number, even in the control animal. But I think, let me see. We have some newer, so we're definitely doing more of that kind of study for the psychedelic, but with a slightly different design. So we can see if we can try to avoid that in the future. But it's actually not that uncommon. If you look at some of the other experiments looking at frontal cortex with 2-photon imaging over time, often you still see some decrease. In fact, we cited some of those papers. They just don't mention it that much.
Okay, so we are out of time. Thank you so much, Dr. Alex, for joining us today, and thank you for such a wonderful and interesting talk.